Sumeet studied physics and optics at the Indian Institute of Technology in Delhi, sorry. And he worked then as a Marie Curie research fellow at the Charité Berlin and with PicoQuant. So PicoQuant is also located in Berlin. And he primarily focused on his research work was to develop spectral resolve fluorescence lifetime imaging, so spectral FLIM systems corresponding to data analysis algorithms. Yes, and he will speak about spectral film, so multiplexing film. Yeah, what he did together with the group from Andreas Hocke at the Charité Berlin, and maybe very short. So Andreas Hocke, I just met once before, but I guess he was was busy with fighting Corona at the moment. So hopefully he can do there something. So Sumeet, I'm really looking forward to your talk.
Thanks Thomas for the nice introduction. So I've been given the task of holding the crowd after 5:30, and even the tougher task to give a talk after this time. But let's see how it goes. So I'm Sumeet. I'll be talking about the recent developments in spectrally resolved fluorescence lifetime imaging microscopy and how we can expand the number of labels using cross-labeling antibodies and FET in specifically directed towards increasing the number of labeling targets in human lung tissues. Most of the work was done at PicoQuant and Charité. So it was a very nice collaboration between the two. And I'm pretty sure there are a lot of, or maybe at least some colleagues from PicoQuant present in this talk. So before I even go to my talk, I would like to thank at least all the senior scientists and my professor from Charité, and of course, the large part of it was from the funding by Bitmap, an initiative of EU Horizon 2020 program. And my special thanks to Katerina, because she was the person who was mostly responsible for the preparation of the samples. And a huge shout out to...
We can't see your presentation yet.
So,
excellent.
Yeah, excellent. Okay, cool. Thanks for noting down. Okay, so these are the people who have been really helpful and immensely grateful to me and helping me out in my PhD time. And moving on, so today I'll be presenting what is the problem of cross-labeling secondary antibodies and what consequences it leads to specifically in multi-target labeling in human lung tissues. And briefly, I will go over the experimental setup which was used to acquire the data, and then moving on to the data analysis algorithm, which is called pattern matching algorithm. Basically, it's a linear unmixing algorithm. I will give a small detail on this part as well. And then we will look at some of the application of the spectral FLIM or SFLIM setup. So first we will look at the cross-labeling, how we can separate the cross-labeling antibodies in cells. And then we will move on to its application in staining multiple targets using limited number of fluorophores in human lung tissue samples. Before that, we will just look upon how we characterize the autofluorescence using the spectral FLIM setup. And if you guys are brave enough and you stay until the end, I will give you some preliminary results from a new FLIM analysis, you can say. So first, let me state the problem straight away. What I would like to show here first is what is non-cross labeling before I move on to cross labeling part. So what we see here is that we have two cellular species called pancytokeratin and Tom20. So Tom20 is shown here in green, and in red is pancytokeratin. So you can see that pancytokeratin is omnipresent throughout the cell. And the way we label them was first we use primary antibodies. Mouse goes to pancytokeratin and rabbit primary antibody to Tom20. And it's a sequential step of immunolabeling. So, and then comes our secondary antibody, which is also fluorophore tagged, Alexa 555, and goat anti-mouse goes to pancytokeratin, and then comes the secondary antibody for the Tom20. So we have a clean labeling of green dye onto Tom20 and red dye onto pancytokeratin. And when we perform our conventional confocal channel mode imaging, we can easily separate these two species because we don't have any cross-labeling. So now we come to the point, what is cross-labeling? So let's assume a hypothetical situation where the primary antibodies are the same on both the species, but let's say by accident or by unfortunately, we have only rabbit anti-mouse present for the pan-cytokeratin. So you perform your immunolabeling step as you did in the last time. But when you go on to performing the immunolabeling for the TOM20, we have the 488 code anti-rabbit, which will also go and stick onto the rabbit secondary antibody, which is present on the pan-cytokeratin. So now we have a cross-labeling of 2 dyes on pan-cytokeratin and TOM20. So when we perform our channel mode imaging, what we can see here is a presence of a false positive signal of pan-cytokeratin in the green channel. And this is difficult to separate because now there is a spectral overlap. So channel mode imaging is insufficient in this case. And the problem becomes more severe when we go on to the multi-target labeling. So let me present a scenario here. So we have, let's say in the lab, we want to label 5 antigens, antigen 1, 2, 3, 4, 5, and we want to label them using different fluorophores. But the problem here is that the well-performing primary antibodies for these antigens, they come from the same species. So as you can see here, they're for antigen 1, for antigen 2 and antigen 4, we have the same primary antibodies. And then when you go on to labeling using your secondary antibodies, which attack with fluorophore, we get cross-labeling onto antigen 2 and antigen 3. So we see that there will be false positive signals coming from antigen 2 from this dye, which is LXO594, and the similar scenario for antigen 3. So we have cross-labeling present on 2 antigens now. So, and on top of that, when you are doing immunolabeling in lung tissues, we have the problem of autofluorescence, which is sometimes even at the same intensity levels as these dyes. So the problem is twofold here. We have cross-labeling and the tissue autofluorescence. So what if you flip this question around and say, okay, hey, can we use this cross-labeling of dyes, which leads to the FRET because they have the right conditions to go under or exhibit FRET. So can we use this FRET to create new fluorophore channels or the pseudo color channels, and we can separate the signals coming from all these 5 antigens. So as Professor Esposito already gave a nice primer on FRET, so we know that, okay, the FRET leads to a reduction in the intensity of the donor emission and the reduction in the apparent lifetime of the donor itself. So we have a combination of things going on here. So change in spectra as well as a change in the lifetime. So standard stuff on fluorescence. There is definitely coming from the last slide, we see that there is a definite need for the multidimensional characterization. So we have 3 different ways or let's say 4 if we count the polarization as well. 3 different way of acquiring the fluorescence signal. So one is the channel mode imaging, where you can just create a few channels, and then if you have nicely spectrally separating dyes, you can, or just use filters to avoid the spectral bleed through. You can separate the fluorescence from the 2 species, and then we have the multispectral imaging, which is commonly used nowadays, especially for multi-target labeling. And then we have the fluorescence lifetime imaging, where we can just acquire the TCS-PC-based FLIM data. So, but as Professor Esposito said that we can, he calls it hyperdimensional, but I call it multidimensional because we are not adding the polarization dimension to it. So let's say it like that. So we can combine all these different mode of imaging and come up with an experimental setup called a Spectral FLIM or SFLIM. So let's look at the setup here. So the setup was completely custom built at the PicoQuant. What we have here is that, especially in this research work, we only use 2 laser lights, so 485 and 560. So this to shine the sample, and the backscattered light or the fluorescence light from the sample is collected onto the detector via separating the light using a spectrograph into its different constituent colors. So we have 8 channels, 8 spectral channels, and each has a channel width of 19 nanometer, and they cover a spectral range of 500 to 640 nanometers. So a min max of 140 nanometer, and we have the maximum efficiency of the detector at 45% at 540 spectral wavelength. And then we have 8 channels. There is a one-to-one topology between the spectrograph and the Hydra hub, which is a PicoQuant product. And what we get here is a 3-dimensional data structure here. So we can multiplex using different lasers. So you can add more laser as well, but we have here only 2. So for each wavelength, what we get is the fluorescence lifetime or histogram, and each of the wavelength or the spectral pin. And just to say that the minimum time bin width of the TCSP series is 4 picosecond, but I believe that there is now a new product with 16 channels, and I think it's higher resolution and even a lower dead time, so you can even go faster. I think it's called Rapid FLIM. So all in all, we have this beast of a system here, and let's see what we can do with it. Okay, so we come to part of separation of cross-labeling antibodies. So before we even translate this idea onto the cells, we wanted to verify that is it really true that what we assume here, that the secondary antibodies which attacked with fluorophores are undergoing FRET or not. So we performed a lifetime measurement. So we have 2 scenarios available here. One is the non-cross-labeling pair. So the pair are red and green dyes. So they don't bind with each other because of their secondary antibody type. And in the plot here, we see that the x-axis is the time frame of the experiment, and on the y-axis we have the lifetime. So exactly at 60 seconds, we mix one of the dyes. So already in the solution we had Alexa 488, and then we add Alexa 555, and there's no change in the lifetime as these 2 dyes are mixed. So it shows that there was no cross-labeling occurring because there was no reduction in the lifetime. But if you move on to another scenario where we have cross-labeling FRET pair, we see that we have the cross-labeling secondary antibodies, and at the moment you start the moment you start mixing the 2 dyes, we have the reduction in the lifetime of the donor, and then this is exhibited by the green trend line here. So this points out the fact that indeed the secondary antibodies with the appropriate fluorophores on attached to them, they do undergo FRET. Then we translate this onto the cell, and again we come to the point where I want to demonstrate that it is difficult to separate using the confocal channel mode imaging. So we have the same thing which I showed before. So in green channel we have a signal from Alexa 488, and then the cross-labeling signal on the pan-cytokeratin, which is shown here in the arrowhead. And this is difficult to separate using channel mode imaging. So then to remedy or to mitigate this issue of cross-labeling, or to discriminate the signal coming from single label and cross-label species, we use the spectral FLIM setup to acquire the data, and then we analyze the data using the pattern matching algorithm. So what the pattern matching algorithm does is basically it takes in reference pattern from single labeled TOM20 and the cross-labeled pan-cytokeratin, and then we perform some unmixing, and what we get in the end is the quantitative separation of photons for TOM20 and pan-cytokeratin in 2 separate channels. So this is quite nice that now we can use a multidimensional setup to separate the signal from single and cross-label species. Okay, so far we saw that we can do it for just 2 species, but there was no fun in it. So let's see, we extend the problem to labeling 3 cellular structure in A549 cell line, just using the 2 fluorophore. So the trick here is again to use a sequential labeling step. So so first we label the primary antibodies onto the pan-cytokeratin and TOM20. Then comes the secondary antibody for pan-cytokeratin, and then we have the secondary fluorophore tagged secondary antibody for TOM20. So this 488 goat anti-rabbit will now go on to pan-cytokeratin as well, and then we have the cross-labeling of pan-cytokeratin via the 488 and 546 dye. And then comes our third cellular species, and it's mouse anti-Golgi primary antibody, and then comes finally the rabbit anti-mouse secondary antibody for the third species Golgi. So this 546 will also bind on to, since it's a rabbit anti-mouse, So it will also bind onto the primary antibody of pan-cytokeratin. So we have the enhanced FRET signal on pan-cytokeratin in this scenario. So it should be a bit more easy to separate. So here in the right side image, we see a fast fluorescence lifetime image. So it is easy to figure out what is TOM20 and pan-cytokeratin, but we don't see much signal from the Golgi apparatus, which is very close to the nuclear part. But we can definitely do spectral FLIM to the rescue, and using a combination of spectral FLIM and pattern matching algorithm, we can easily separate the contribution from the single and cross-label species into different channels. So on the top hand side, we see that using channel mode imaging it's really difficult, even if you use smart filtering techniques or the bleed-through cancellation techniques. And using SFLIM, it's pretty easy. You just find out the reference patterns. So in this case, the reference pattern for the Golgi was created using a single slide. So by single slide, I mean a separate slide was created for just with the labeling of Golgi with 546. So we take the reference pattern from there. But for the pan-cytokeratin and TOM20, we can easily find pixels which gives the reference pattern for TOM20 and pan-cytokeratin. And then we perform our usual pattern matching and find the unmixing find the contribution using the unmixing algorithm. So here we can nicely see that they're all separated into their constituent independent fluorescence channels. And so all this data which I'm presenting here has already been published. So during the review, one of the reviewer asked, like, can you show like really in a quantitative manner or in some visual manner how spectral FLIM is better than channel mode imaging? So the idea was just to use a line profile. So you just draw a line and then you find out the contribution or the intensity contribution from different species. So for channel mode imaging, you see that there is always some contribution from pan-cytokeratin into TOM20 and Golgi channel. But after unmixing, we see that as in the pink is Golgi. So it's nicely just along this line, we find the contribution alone from Golgi, which is not the case here. You see the bleed through of pan-cyto into the Golgi channel. And the similar thing you also observe in the green channel of TOM20. So you can see that they are nicely separated. So if there is some young PhD student and if you're looking for some way to prove some points, maybe you can use this kind of a chart here. So this is what I wanted to point out here. And coming to the pattern matching algorithm, originally it was developed by the group of Jörg Enderlein and specifically Ingo Gregor. And thanks to him for providing this algorithm. What I did during my PhD time was that I improved on this algorithm. So by improvement, I mean the speed improvement. So the first one I have shown you already, that we use multi-core processing to speeden up the image processing. But then we thought, okay, is there any other way to speed this further? Because usually without any speed improvements, it was taking around 2 hours to analyze the data for a single image of 512 by 512 pixel. And then when we used the multi-core processor, the improvement was around 16-fold because there were 16 cores. So we came down to time of around 10 minutes or something. But then we wanted to improve it even further in order to optimize on our experiments. Then we used some techniques from deep learning. So the technique is that you just use some momentum-based speed up. So in what we see here, let me guide you through, is the aim of the pattern matching is to find these A1, A2, A3. These are the abundance coefficients corresponding to 3 different patterns. So in this simulation here, the coefficients were set to 1500, 800, and 7000. And then using this momentum-based speed up, what we see here is that the original algorithm, which is shown here in blue, is taking it will go on to around 1800 steps or something. But using this momentum-based technique, we can bring it down to 400 steps of iteration. So this is huge improvement of around factor of 4. And this is something which will be published soon also. And this I think already released in the open source on my GitHub. So if you are interested in that, you can definitely use it. So from 2 hours, we have come down to now 2 minutes of analyzing a single image, so which is a huge improvement, I would say. and makes the life of the researcher quite less cumbersome. Then come to the point that we wanted to translate this trick of cross-labeling onto the human lung tissue samples. So before that, as I mentioned, that autofluorescence is a huge roadblock. So we wanted to characterize the lung tissue autofluorescence as well using the spectral film. And to emphasize more that why the autofluorescence is such a showstopper in lung tissue imaging is that you can see here that that using different excitation, what is shown here is that we have different excitation on the same tissue sample. So 405, 488, or 561, and the spectral profile, it's all over the place. So it covers the whole visible range. So the point here is that there is definitely a huge contribution towards the data, or towards the data in terms of autofluorescence, even though. Okay, yeah, no. So let's move. So how do we characterize the autofluorescence using spectral film? So in order to incorporate a large amount of data, what we did here is that we used tissue samples from 20 patients, and per each patient we imaged 3 different regions of interest. And out of these 3 images, each of the image was used to extract 3 distinct patterns. So 3 distinct patterns correspond to the red blood cells and the collagen and the cytosolic, which is mostly the background autofluorescence. The way to, so one would ask, like, how do you find this region? So the easiest way is that we take a film image, we make a decay diversity map. So decay diversity map is a plot where we plot where on the x-axis we see that is the average lifetime within 1 pixel, and on the y-axis we plot the variance of the lifetime within that pixel. And then nicely it comes out that there are 3 different clustering regions. So using these clustered regions, we can select these pixels where the contribution from the single species is there. And what comes out is that we see here that in the spectral profile, the spectral emission of these 3 different species is not that distinct. But when we come to the lifetime part, we see that for the 485 excitation, they separate nicely. So this is already a nice indication that adding the lifetime to your data analysis adds some information, and I will show you how. So on top row, we see the spectral based unmixing only. So only spectral information was used. And in the bottom row, we see the S-film based unmixing. So lifetime plus spectra, both information were used for unmixing. And then we see that erythrocytes, there are some regions where we have a strong bleed through of erythrocytes into the cytosolic region or even collagen regions. But when we use the spectral or all the information available to us, the bleed through is reduced from 14% to 4%. So we definitely improve our data analysis part. So meaning that we can find finally separate the quantitative contribution from 3 different species. This will become important soon. So application 3. Now we translate this cross-labeling antibodies to label 3 different antigens in the human lung tissue sample. The way we did it is, SPC was labeled with 488. So if we will soon release the details on this experiment as well, but let me explain it here as well. So we have 488 on SPC and 546 on CD68. The way the immunolabeling was done in a sequential way, we ended up with the combination of, or the cross-labeling of EMP with 488 and 546 dye. And on top of it, we also had the presence of autofluorescence. Since we already characterized the autofluorescence, so we could really assign the reference pattern to these 3 autofluorescence species. And then using the reference slides, we created the reference pattern for the SPC, C68, and EMP, just to be on the safer side. Although we could have chosen it from the image itself. And now when we do the unmixing, you can easily see that how well separated they are, even though in the presence of autofluorescence itself. And this is something really nice. And in the human in the lung tissue community, this is always a problem that, especially in infection studies, you cannot label multiple antigens simultaneously because of this cross-labeling issue. So now this kind of a trick allows you to label multiple antigens at the same time and visualize them in a quantitative manner as well. So before leaving the lab, I had the opportunity to even push or test the setup, how far we can go. So here what we have is the lung tissue is labeled with the opal dyes, which are the new dyes. So they have really nice characteristic, distinct or like single spectral line in the emission spectrum. So opal 522 is labeling MERS coronavirus, which is always affecting or on the outer lining of the samples of the lung tissue samples. And then we have SPC labeled with Alexa 555 and CD68 with opal 620 and EMP with opal 540. So they are really spectrally very close, especially these three fluorophore are spectrally very close to each other. So adding lifetime definitely benefits the whole experiment here. And then we can nicely see that which are the regions where the MERS coronavirus is affecting the tissue sample. Yeah. Okay, so far what we have seen is that cross-labeling not necessarily is a huge problem, but it can be turned around and we can use it to potentially expand the number of possible target labeling at no extra cost, because all you have to do is perform some really nice, strong control sample so that you know what is going on. So if you have any background signal coming from the extra concentration of the dye, which was used to create the cross-labeling, and you also have to perform some control samples where you know that where only the donor dye is present on the cross-labeled antigen, so that you know what is the donor lifetime in absence of acceptor. So all in all, what it needs is a smart selection of interacting fluorophore tags on the cross-label antigen, and this only becomes possible by use of spectral FLIM. Scalability is very nice to have. So in this setup, you can definitely multiplex more laser lines, and you can expand the number of channels. As I said, that now there is a system available with 16 channels itself. And if you want, and if it's required, you can also add the polarization part to it, like Professor Esposito's setup does. And we have the 2D data structure available, which is a combination of spectral and lifetime information. And if you choose your reference pattern nicely, then you can discriminate between the different markers, even though they have the similar spectral properties very easily, even in the presence of tissue autofluorescence. And as I said, that is definitely a mandate to select your reference pattern in a very nice manner. And by use of pattern matching algorithm, now it's improved further, so you can definitely have a pseudo online analysis of your data. So all in all, what we saw is that spectral FLIM definitely allows you to significantly improve the identification of multiplexed markers in the complex tissue samples, even in the absence of the strong autofluorescence signal which is present in samples like lung tissue. So moving on, since you guys are still here, I want to present you an idea which I've been working in the last 1 week. And the idea is like this. First, the problem statement. So what we have here is from left to right, we have number of photons and the histogram for corresponding number of photons. So we have 1,000 photons, the histogram corresponding to that. And then we increase to 10,000 photons, 30,000 photons, and then 100K photons. So this is what the evolution of histogram looks when you have increasing number of photons. So using limited number of photons, let's say we have so many times in FLIM imaging, we have scenarios where you can acquire only limited number of photons, but then you would like to have more photons in order to do a very stable lifetime analysis. So can we use deep learning methods to learn the underlying pattern for photon generation, let's say using 1,000 or 10,000 photons? So this is very basic study which I did in last 1 week. Let me show you some results. So first experiment, what I did is just using first 10,000 photons. Of course, in deep learning, you need a lot more data. So this was my first go-to experiment. So I'm not sure how many of people have looked at the raw TC-SPC file, but this is how it looks like. So we have photons coming on, and each photon is assigned a TC-SPC bin. So in this case, we have bins going to 2000. And so this is the evolution of your experiment here. So this is what the histogram looks at the 1000 photon mark. And then if you include more photons, the signal becomes better. And then you can perform your lifetime exponential fitting analysis and whatever you like. So we can formulate this problem as a time series regression and use a deep learning framework or a network to learn this underlying pattern and generate, can we go from here to here? So prior work has shown that long short-term memory networks do have the potential to learn this kind of a pattern, and they do have a good inductive biases. So without further ado, what I did a very quick setup of experiments. So I take the first 10,000 photons. This is my data set, train an LSTM model, and then use this model to infer the next 100K photons here, and then build the histogram. So the data set, the preliminary data set looks like this. Then we generate the next 100,000 photons. So please don't be fooled by the scale, because this is 10 to the power 2, and here is 10 to the power 3. So they are definitely different. And this is what was expected. So definitely it looks a lot more noisy, but something tells me that this is definitely possible. So this requires further deep dive. So one way to verify would be to fit a lifetime and look at the prediction errors and so forth and so forth. And if a young PhD is interested in taking up this research further, I'm definitely willing to let you have the scripts and ideas, and if you need more help. Since I don't have time, that's why I'm letting go of this idea now. And so LSTMs come in different flavors. So what I implemented or showed you here was a regularized LSTM. So it comes from Stephen Meredi. So he came out with different ways to regularize your networks. Then something which I think can really work for low number of photons is attention-based models. And this is something to look really closely for. And maybe LSTM on autoencoders can also work. So I'm just letting you like different directions which you can explore, given you have time or you are in your first year of your PhD and you don't know what to do. So some practical advice would be just to use a very nice cross-validation data set to find optimal hyperparameters, because I did not have time. So I just brute force this without a cross-validation data set. And the network is extremely fiddly to train. So be careful when you see something and you don't understand. So it's really important to have a firm understanding of how LSTMs work. And the next step would be to lose low number of photon and then infer the next 100K photons and build your histogram and fit the lifetime and see if you can come up with a good inference model based on low number of photons. So the far-fetched idea is a multi-time series regression. So again, I'm not sure how much you have looked at the raw data format of FLIM image itself. So it contains spectral channels, TC-SPC bin, and line numbers. So line scan. So if you have 512 by 512, then it will range from 1 to 512. So we have 3 different time series going on. So maybe you can use a multi-series LSTM to do this kind of inference and have more pseudo virtual photons to your image to aid your image analysis. Okay, so I think that's where I would like to end, and I'm willing to take questions now. Thanks for your attention.
So thank you, Sumeet. How do I pronounce it correctly?
Yeah, it's the second time you pronounced right.
Okay, perfect. So thanks. Yeah, there was quite a lot of discussion coming up in the chat, especially when you started with the autofluorescence part. But there was one question just right at the beginning from Alessandro.
Ah, yeah, okay.
He directly asked, so basically, when can I buy your detectors?
Alessandro, you know who to ask. Don't ask me. I'm not the representative here anymore.
Okay. Yeah, so let me see. So the next question is from Nguyen. And he is asking you, I guess this was also from the autofluorescence part, if you can do similar separation or unmixing things without your special detector. So I'm not sure how many detectors he have at his setup, maybe 2, maybe you can unmute yourself, because he just has a normal TC-SPC setup. So is this then also possible to do this?
It's very nice that you ask, because in the beginning of my work, I exactly did this. I had a confocal setup with 2 channels only and lifetime added to it. It definitely improves the quantitative separation. And what I've seen is that as you add more spectral channel, it definitely improves it even further. So adding lifetime information always helps. Yeah.
Okay, so the unmixing would work. But not as good as if you have this multi-detector.
Yeah.
Okay. So in a similar direction, from Hella is asking if 4 spectral channels might be useful for addressing autofluorescence already, or if this is too poor, but I guess it's
4 is enough. I've done some simulation where I crunched down, like added 2 channels together because I had 8 channels. So I just combined 2 of them together. So just add the data, just additive operation, and then it works the same way as you would have 8 channels. But yeah, for autofluorescence, it works nicely. But when you want to have multiple fluorophores as well with the autofluorescence on top of it, then it adds more information to add more channel as well. I think I want to point out to the community here that Professor Esposito wrote a very nice paper about this. It was about I forgot, Fisher information maximization. So I think this would be something, a very nice direction to also go in, like how much information is enough to do the unmixing using the Fisher information as a metric there.
Okay. So we will write this down maybe. So the next question from Dovidas. In your opinion, would it be possible to use antibodies for protein interactions experiments via FLIM thread? So instead of using fluorescence proteins, that you're using antibodies, and maybe if the size of the antibody hinders the thread efficiency. So is it better then to try nanobodies because they are smaller if there's a difference in your thread efficiency?
This is exact. Okay. Professor Esposito is pointing out that he's not professor, but in my eye, you are. Okay, coming to the question that definitely, as you point out, that the size of the antibody does play an effect, and there is already a nice paper which states that you should not use the full IgG molecule because it hinders the interaction between the secondary and the primary antibodies. So this would be something again to try it out, because I'm not a sample preparation expert, but with my minimal knowledge, this is what I can say.
Okay. So Stephanie Weidkamp-Peters asked if it would make sense to share reference signals among labs, so that you have different references available.
This again is a very nice idea. I think a couple of labs can join their hands and they can generate a reference library. But the critical point here is that the sample preparation protocol should remain same, at least for the cellular species or the tissue species which you are labeling to. So if the sample preparation steps change, then the environment will definitely change the fluorescence lifetime properties or the spectral properties of the dye, and then your the new reference spectra might differ from the one you have in the library itself.
So we would need a protocol. And maybe a control that our protocol is working in 2 labs the same.
I can tell you even the concentration affects the reference pattern. So you have to be really critical there.
Mm-hmm. Okay. So then there was one question. Wait, I'm missing it. From Sandra. I guess this was coming up during the deep learning part of your talk. If not the reference patterns you used there differ from different cell types, and I guess you.
I didn't get the full question, I think.
So wouldn't the reference patterns differ within different cell types?
Yeah, so then you will have, so this is the advantage of using low number of photons. So let's say you can train a network on the fly using just 1,000 photons. So it just takes few minutes to train a network like this. Yeah. So you can definitely train a different network for different cellular line or different reference, different antibodies and stuff like this. This is really like last 1 week of work, I would point out that be careful when you try out this stuff. You might lose a lot of time in your PhD doing this.
Okay. So thank you a lot. Sometimes great talk. And I guess there's one last question also from Sandra. Would you expect different patterns for the same sample at different systems, even under identical imaging conditions?
It would be a stroke of really bad luck, I think, if that is supposed to happen. But that can happen for sure.
And maybe I have one question. Have you ever compared this? So the first part of your talk was if you have the same antibodies. So they are coming up these nanobodies where they claim that you can pre-mix them, that you can stain against the same host. So have you ever compared this to your technique, and is it working? So I haven't tried it yet.
No, no. Personally, the problem is that with the human lung tissue samples, anything, if you want to try it out, it takes 6 months to find out even if it's working or not, because most of the time you are unsure if you did the right thing or not, right? So even with opal dyes, it took around 6 months to find out that they don't work as nicely as they are promoted in the catalog. Yeah. So if you have time, go on, perform control samples, yeah, please.
Okay. So are there more questions also?
Feel free to reach out via email or Twitter, or if you want the deep learning slide, just take it and do the research. I mean, this was the idea. I learned deep learning on my own on using open source. So this is why I'm letting go of this idea now, because I don't have time to work on this.
Okay. But for Spectral FLIM as well, I do not understand this.
There's a different conversation going on.
Oh, okay. Okay. So then thanks again for the talks. So thanks to all of our speakers today. So to Stephanie, Wolfgang Peters, Yvonne Stahl, Carsten Grashoff, Alessandro Esposito, and Sumeet.